Okay. Thank you also for the introduction. The motivation to this research that I'm going to present to you today is the fact that egg-laying hens have osteoporosis and bone fragility, and this is a significant welfare concern in the commercial egg farming industry. And the cause of this fragility, of course, is multifactorial, but it has to do with the fact that the birds are often housed in cages, and cages do not allow for sufficient physical activity, which is necessary to maintain bone mass. So you get this situation where bones are subjected to below normal mechanical stimuli, and then they end up in bone resorption state. And so efforts to address this have looked towards housing the birds in more spacious environments that give them more opportunities for physical activity to improve their bone health. And it has been shown in some initial studies by Dr. Wadowski's group, where they showed that specifically early life experiences of physical activity had long-lasting benefits to bone health. But these studies were limited in the fact that they looked at one genetic strain of chickens, and they compared birds that were in cages and those that were in one specific style of commercial rearing aviary. But we also know that different genetic strains are physiologically and behaviorally quite different. And what Dr. Wrench showed was that there was a genetic strain-dependent effect of aviary style on the amount of physical activity that these birds were doing, where the white birds had performed more physical activity in moderate and high complexity aviaries compared to low complexity aviaries, but the brown birds had similar activity levels. So based upon this and based upon what we know about how physical activity affects the bones, we thought that we would see similarly a genetic strain-dependent effect of aviary rearing on bone microstructure. But then when we looked at this wasn't the case. So we did see that there were some genetic strain differences in bone microstructure. This, for example, here we have cortical thickness and cortical area fraction, but we actually did not see any differences between genetic strains in terms of the effect of housing. And we also didn't see any significant differences between these aviary styles. So what all of this together suggests is that perhaps there are differences in the way that the bones are interpreting these mechanical stimuli and responding to them, differences between the white and the brown feathered birds. And so this is what I aimed to look into by doing a controlled loading study. So I sampled birds, Leghorn selected, Lohmann selected Leghorn light and Lohmann brown light genetic strains, so a white feathered and a brown feathered genetic strain, which were reared in either conventional cages or two styles of commercial rearing aviaries, one of low complexity and one of high complexity. And these birds were reared at the University of Guelph by Dr. Tina Wadowski's group. And then we brought them over to McGill and did what we call a controlled loading study, where you subject the birds to a loading stimulus that is controlled and that is of a magnitude that is above what is normal, such that it hopefully elicits a bone formation response, and then you can quantify and study this response to this controlled stimulus. And in this case, we applied a force of 180 newtons because I've shown in a previous study that this amount of force produces mechanical stimulus at the tibiotarsus that is 3 to 8 times what occurs during locomotion. And we used an axial compressive loading model. So the force is applied along the long axis of the tibiotarsus such that it mimics what is the loading that is happening in vivo. And this is just a little video of a chicken. And you can see that one limb is being subjected to this intervention where the other limb serves as an internal control. And so initially we decided to look at bone microstructure at the tibiotarsus midshaft, comparing the loaded and the non-loaded limbs. And what we saw, which was really surprising and contrary to what we hypothesized, was that the limbs that were loaded had inferior microstructural characteristics compared to the loaded limbs. That being said, we did see that there was a genetic strain-dependent effect of this in vivo controlled loading, where the white birds were responding and the brown birds weren't. So In another set of data, I modeled the mechanical stimulus that we're applying to the bone along the length of the bone. And you can see that at the midshaft here, which is where we initially quantified the mechano response, there's not such of a high stimulus. But then in these more distal regions, in the proximal and the distal metaphyses, you see higher mechanical stimuli. So we decided that we needed to go back and we needed to reassess the effect of loading in this region of high stimulus. And this is what I'm going to present to you today. So the aim was to, again, evaluate the effect of in vivo controlled loading on the microstructure of the tibiotarsus, this time at the proximal metaphysis, and to determine whether, again, the mechano response is influenced by genetic strain and rearing housing style. And I hypothesized that we would see what we would expect, that the loaded limbs would have enhanced tibiotarsal microstructure compared to the non-loaded limbs. So I imaged the bones again at this proximal metaphysis region. I segmented the images into cortical and trabecular bone to analyze these bone compartments separately. And then I used the PROC-GLM and SAS with repeated measures ANOVA to test fixed effects of genetic strain, housing, and loading, which is limb, and the two-way interactions of these two. And then differences between the loaded and the non-loaded limb was assessed by paired t-tests, whereas differences between genetic strain and housing groups by unpaired t-tests with 2 key post-hoc corrections. And so now here are the results. And just to orient you, we have the different experimental groups, and in white is the non-loaded limb, and in blue is the loaded limb. And below I have this, wherever the paired t-test between the loaded and the non-loaded limb is significant, you can see this arrow, and the arrow indicates how the loaded limb compares to the non-loaded limb. So what's really evident is that similar to what we saw at the midshaft, in many of these parameters, we see that there is impaired mode microstructure in the loaded limb compared to the non-loaded limb. But we do see some positive effects. We see that there's improvement to cortical area fraction here, and also we see that there's a decrease in porosity, which is positive. So decrease in porosity and increase in cortical area fraction are both bone strengthening adaptations. And then we also see here that there is an influence of genetic strain and housing on the effect of loading. In terms of the trabecular bone, we see that there's much less of a response. Sorry, and I forgot to mention that unlike in the midshaft, here we do see that not only the white birds, but the brown birds are also responding to this mechanical stimulus. In the trabecular bone, however, we see that there's barely a response, as seen by no significant loading effect in loading ANOVA effects. But we do see that, interestingly, all parameters were influenced by rearing housing. So that's something to further look into. And so main findings are, first of all, we saw this really striking regional difference in the effect of loading, where the cortical bone was quite influenced, but not the trabecular bone. We saw that in both the midshaft and the metaphysis now. In vivo loading led to impaired bone microstructure, but at the metaphysis, we did see some improvements, namely in cortical area fraction and cortical porosity. And now, in line with my hypothesis for this study, we did see that there was a response to this intervention in both the white and the brown feather genetic strains. So the next natural step is to try and really understand what is behind this response, is to look at the cellular behavior, the behavior of the cells that are carrying out this response, namely bone forming cells and bone resorbing cells. And so we can answer this question, well, are these negative effects of loading, is this due to decreased bone formation or increased bone resorption, or maybe both? And so this, we're looking at it now with histological approaches. And then I'm also looking at doing this same thing, but in this distal metaphysis region, to see if the results agree with what I've showed here in the proximal metaphysis. And something that I won't get to, but probably is really interesting is to look and see if maybe we're applying too much force and we're causing micro damage, and then you have a whole different healing response that is separate to this healthy bone anabolic response. So with that, I'll finish, and I'll just thank our collaborators, Dr. Wadowski, Dr. Wrench, Dr. Main, and my lab, and of course the funding agencies. Thank you.
All right. Thank you very much, Isabela, for a great presentation. We have time for just one quick questions, and I'll take it from the Q&A from the chat. So the question is, what would you do differently if you could redo your experiment, maybe in view of kind of the results that were kind of unexpected or unanticipated? So what you think would be done differently if you had the opportunity?
Well, yeah, that's a great question. So in our experiment initially, we wanted to have a controlled experiment where we're applying the same amount of stimulus to both genetic strains. And so what we did is we did some initial measurements of this mechanical stimulus, and we did it here at the mid shaft. And as you can see, the mechanical stimulus is similar between genetic strains here. But then when we did some simulations, we see that in these areas where the stimulus is higher, there's also a difference between white and brown birds. So this means that our experiment is not perfectly controlled in these sites of the bone. So definitely if I could go back, I would repeat it where I'm applying lower forces in the white birds compared to the browns, such that these forces are producing similar strain stimuli across both groups. And yeah, that's what I would do.